Imagine for a moment that every single living thing you have ever seen, trees, birds, dogs, people, was once nothing more than dust. Not just metaphorical dust, but real particles, scattered across a young earth that was boiling, violent, and utterly lifeless. What if I told you that inside your body, right now, there are atoms older than the earth itself? Some of the hydrogen in your blood was born in the core of stars that died long before our planet was even formed. And yet somehow, here you are, not only alive, but listening, breathing, and perhaps blinking slowly as you try to settle into sleep, while still curious about how you exist at all. Welcome to Science for Sleepy. Tonight, you and I will quietly unravel what science currently understands about life's beginnings. And before we sink into the details, can I ask you for a small favor? Could you head to the comments and let me know where you're watching from and what time it is there? It's a strange kind of magic, really me speaking here, and you listening perhaps on the other side of the planet at some odd hour. And while you're there, please subscribe to the channel. Think of it this way. Subscribing is like the chemical bonds that hold life together. Without those connections, nothing sticks and everything falls apart. We post a new video every single day, so when you wake up tomorrow, you'll find a fresh story waiting, ready to carry you back into science at its sleepiest pace. Now, let's return to the beginning not of this story, but of all stories. Earth, 4.5 billion years ago, was unrecognizable. Forget the soft blue oceans, green forests, and buzzing insects that you know today. Instead, picture a cracked, blistering surface, hammered by asteroids, volcanoes belching gases into an atmosphere that would have suffocated you instantly, and oceans that were not yet oceans as you understand them, but broiling chemical soups. And yet, this chaos held the seeds of everything to come. Scientists often remind us that life leaves behind fossils, but the earliest life did not have bones or shells to fossilize. What it left instead were traces, whispers in the rocks, chemical signatures so faint and so dull that you could walk past them and never notice. The oldest of these whispers date back to more than 3.5 billion years ago. Imagine that for a second. We're talking about forms of life that existed before continents were stable, before the moon had settled into the orbit we know today, before even oxygen filled the air. But how did lifeless matter cross that line into life? It wasn't a single dramatic moment, no lightning strike in a laboratory that suddenly created a walking creature. Instead, the origin of life appears to be a slow choreography of tiny, almost boring steps. Molecules bumping into each other, reactions forming chains. At first glance, it seems dull, but the dullness is the wonder. Imagine watching raindrops slide down a window pane. They merge, separate, and sometimes form larger droplets. Now replace those drops with the earliest molecules, simple carbon compounds, forming slightly more complex arrangements. Nothing exciting at first, but given enough time, those arrangements learned how to copy themselves. And that, in essence, is where life truly begins. The difference between a random chemical and something that insists on making more of itself. One of the leading ideas in science is known as the primordial soup hypothesis. It suggests that early Earth's oceans were filled with simple molecules, methane, ammonia, hydrogen, and water vapor, that could, under the right conditions, spark more complex chemistry. The famous Miller-Urey experiment of the 1950s tried to test this by simulating those early Earth conditions in a glass flask. They filled it with gases, zapped it with electrical sparks to mimic lightning, and waited. Within days, the flask turned pink, then deep red, as amino acids, the building blocks of proteins, formed. It was shocking at the time. Proteins are essential for life, and here they were, emerging from nothing but gas and electricity. Yet, as groundbreaking as it was, this experiment didn't solve the riddle. Amino acids are not life. They're ingredients, not the recipe. Imagine finding a pile of bricks on the ground. It doesn't mean you have a house. Something more was required, something that could arrange those molecules into self-sustaining systems. Another fascinating clue lies in the discovery of hydrothermal vents on the ocean floor. These vents are like underwater chimneys, spewing hot, mineral-rich fluids into the cold, dark sea. In the late 1970s, scientists exploring the Galapagos Rift stumbled upon these vents, and around them, entire ecosystems of creatures, giant tube worms, crabs, and shrimp that thrived without sunlight. Instead of using photosynthesis, these organisms lived off chemosynthesis, harnessing the chemical energy released by the vents. Some scientists began to wonder, if life could thrive in such places today, could life have begun there billions of years ago? The vents offered a protected environment, a steady flow of chemicals, and surfaces on which molecules could cling and interact. To this day, many researchers believe these dark, alien-looking landscapes might have been the birthplace of life. And yet, another school of thought points to the sky. Some suggest that the building blocks of life may have arrived from beyond Earth, hitching a ride on comets or meteorites. When scientists studied certain meteorites, they discovered amino acids and complex organic molecules within them. Imagine that the same rocks that streak across our night skies, burning out in a flash, may once have carried the seeds of life to our young Earth. If true, it means life's origins may not be confined to our planet at all, but could be a universal phenomenon waiting to happen wherever conditions are right. But here's the quiet, humbling detail that scientists often emphasize. No single theory yet explains everything. Life's origin remains an unfinished puzzle, pieces scattered across disciplines, chemistry, geology, astronomy, and biology, each offering hints, but not the full picture. Perhaps it was a combination, some ingredients formed on Earth, others delivered from space, stirred together in oceans or clung to rocks near boiling vents, eventually stumbling upon that most magical property, the ability to replicate. When you think about it this way, the story of life's origin isn't just about one lucky spark. It's about persistence, repetition, and time. Endless time. Billions of years of trial and error, where molecules collided, broke apart, and reformed, until finally a pattern emerged that was stable enough to last. And once that pattern existed, evolution took over, sculpting complexity from simplicity. As you lie there listening, you might ask yourself, if life began with such simple chemistry, almost like an accident, does that mean life could emerge elsewhere in the universe just as easily? Or is Earth's story a rare, one-time event, a coincidence never to be repeated? If you were to step back into Earth's earliest days, the world would not feel like a place where life could possibly begin. Imagine a sky filled with choking gases, an ocean more acidic than vinegar, and continents still forming, cracking, and shifting. It was not a home, it was a cauldron. And yet, within this seeming hostility, the very first whispers of life were shaping themselves out of chemistry. To understand how that could happen, scientists often zoom in on the tiniest and most ordinary of things: carbon. You carry it in every cell of your body, but it is so familiar that it becomes invisible. The graphite in a pencil, the fizz in soda water, the black smudge left from a candle, each is carbon in a different form. Carbon is not rare, it is abundant across the universe. What makes it special is its flexibility. Unlike most atoms, carbon can form 4 bonds at once, like a hand with 4 fingers reaching out in different directions. This property allows carbon to link into long chains, rings, and branches, creating a staggering variety of molecules. Without this flexibility, life as we know it simply would not exist. Now, when early scientists thought about the first spark of life, they often imagined a dramatic event, a lightning bolt striking a pool of chemicals, or a comet delivering a ready-made recipe. But as the decades passed, the picture grew less cinematic and more methodical. Life did not require magic. It required repetition, energy, and surfaces where molecules could meet. Think of it less as a thunderbolt moment, and more as a slow rehearsal, billions of tiny accidents that eventually gave rise to patterns. Take RNA, for example. You might have heard of DNA, the molecule that stores your genetic code. But before DNA, scientists believe RNA may have played the starring role. Unlike DNA, RNA can not only store information but also perform simple chemical reactions, making it a versatile candidate for the earliest stages of life. Some researchers even call this the RNA world. The idea is that small strands of RNA formed spontaneously on early Earth, perhaps clinging to clay minerals that acted like scaffolding. Over time, these strands became better at copying themselves. Imagine a little sequence of molecules stumbling into the ability to replicate not perfectly, but well enough to survive. Each imperfect copy introduced variety, and from variety, evolution had something to work with. Of course, critics of this idea point out a problem. RNA molecules are fragile, easily broken by heat, radiation, or water. So how could they have lasted long enough to matter? That question remains open, but experiments have shown that under the right conditions, protected inside tiny bubbles or attached to mineral surfaces, RNA can indeed persist and even replicate. It might not have been the whole answer, but perhaps it was one of the first stepping stones. And that brings us to another key concept: compartments. Life is not just chemistry floating around in soup. It is chemistry contained, protected, and organized. Even the simplest bacterium today has a membrane, a thin layer that keeps the inside distinct from the outside. Without that separation, molecules disperse too easily and reactions lose their direction. Some scientists propose that early membranes formed naturally from fatty molecules, which can spontaneously arrange themselves into tiny spheres when placed in water. You can even observe this in a laboratory. Drop a bit of soap into water, and it forms little bubbles on its own. These primitive bubbles could have cradled fragile molecules like RNA, creating microenvironments where reactions could concentrate and repeat. This may sound unimpressive, a bubble, a string of molecules, a faint reaction, but when you look closer, it is extraordinary. Life is not built from rare miracles. It is built from ordinary chemistry, performed relentlessly over vast stretches of time. Picture rain dripping onto a stone. One drop changes nothing. But after millions of years, the stone is worn away, reshaped entirely. The origin of life works the same way. The processes were dull, almost invisible, but they carried weight because of their persistence. The fascinating part is that we can see echoes of these ancient processes inside our own bodies today. The way RNA still plays a role in translating genetic information, the way membranes form spontaneously when certain molecules are present, the way carbon chains provide the backbone for proteins and sugars. All of these are living fossils, reminders that we are built on patterns far older than our species, older than mammals, older even than complex cells themselves. Now, pause for a moment and consider what this means. If the first steps of life were not dependent on rare events but on common chemistry, then perhaps the universe is teeming with similar beginnings. Picture planets orbiting distant stars, oceans sloshing beneath icy crusts on moons like Europa or Enceladus. If the same basic chemistry exists there, and it does, then are those worlds quietly rehearsing the same patterns Earth once rehearsed? Are they waiting, patient as stone, for molecules to bump together and accidentally form the first fragile, self-copying system? And yet, here we confront one of the strangest realities. Despite decades of research, no laboratory has ever fully recreated the leap from chemistry to life. We can make amino acids. We can form simple RNA strands. We can watch membranes emerge spontaneously, but we cannot yet put the pieces together into a system that sustains itself, grows, and evolves. It is like having a box of gears and springs but not knowing how to assemble them into a working clock. The result is humbling. Life's origin may seem inevitable in hindsight, but we still cannot reproduce it with certainty. This gap between what we know and what remains unknown often frustrates people. But for scientists, it is a source of quiet fascination. Each unanswered question is an invitation to look closer, to experiment differently, to imagine scenarios that stretch beyond our assumptions. Perhaps we are missing some chemical pathway that early Earth had but no longer exists. Perhaps life began in environments we have not yet considered deep within rocks, or frozen under layers of ice, or in places so extreme that we struggle to imagine them. When you think about it this way, the search for life's origin is not just a story about the past. It is a mirror for our present curiosity. We want to know where we came from, not simply to solve a riddle, but to understand what life itself truly is. If life can emerge from dust, energy, and time, then what does that say about the boundary between living and non-living? At what point does chemistry become biology? At what point does stuff become self? As you lie back and let this question sink in, consider this. Perhaps life is not a sudden spark, but a spectrum. On one end, lifeless molecules. On the other, fully evolved organisms. And in between, countless stages, each slightly more organized than the last. If so, then the origin of life is not one moment in time, but a long, slow gradient, a path we are still trying to map with every dull but meaningful experiment. And here is where I will leave you for now, with a thought that may linger even after you drift off. If chemistry can slowly become life, then how many other planets, tucked away in the silent corners of the universe, are quietly following the same patient path? When you think about the earliest traces of life, it can be tempting to picture something familiar, a tiny fish, a green sprout, maybe even a microscopic bacterium wriggling about. But the truth is far less dramatic and far more subtle. Life did not begin as something you could recognize under a microscope. Instead, it began as chemistry, leaving behind marks so faint that even today, scientists argue about whether we are truly seeing the first signs of life or just curious accidents in stone. The oldest rocks on Earth are more than 4 billion years old, though many have been melted, crushed, or recycled by geological forces. That makes the search for early life like searching for pencil marks on paper that has been erased, rewritten, and burned. And yet, despite the destruction, certain pieces of evidence remain. In Western Australia and in Greenland, scientists have found rocks containing carbon with a very specific fingerprint. The ratios of lighter and heavier carbon atoms suggest they were processed by living organisms. To the untrained eye, it is just dull black smudges in stone. But to a geologist or a biochemist, it is astonishing. A silent message from organisms that lived 3.5 billion years ago. What were those organisms like? Not plants, not animals, not even the kind of bacteria you would recognize today. Instead, they were simple cells, little more than bubbles enclosing strands of molecules. They lacked complex parts, no nuclei, no mitochondria, none of the machinery you and I depend on. And yet, they did one thing that mattered most. They copied themselves, slowly, imperfectly, but persistently. It's worth pausing here to imagine the scale of time. When I say 3.5 billion years, that distance is almost impossible to hold in your head. If you compressed Earth's entire history into a single year, life would appear in March. Complex animals wouldn't show up until December. Humans? We would walk onto the stage only in the last few minutes before midnight on December 31st. That means most of Earth's history belonged not to dinosaurs, not to forests, but to microbes. Tiny, invisible creatures quietly building the foundation for everything that would come later. What is perhaps most striking is how little those earliest organisms needed. They lived in an Earth with no oxygen, feeding off chemical reactions that seem unimpressive today. Iron, sulfur, hydrogen ingredients you might find in a cave or around a volcano. And yet, with these, they endured. It was boring, patient metabolism, the kind that doesn't make headlines, but it was life. One of the best windows into this ancient world comes from stromatolites. These are layered rock formations built by colonies of microbes, often cyanobacteria, which trapped sediment and secreted slimy films that hardened over time. Imagine a microbial city, growing upward slowly, each new layer covering the one below. Fossil stromatolites have been found that are over 3 billion years old. They look like lumpy rocks, unremarkable unless you know what they are. But once you do, you realize they are skyscrapers built by some of the earliest communities of life on Earth. And even more fascinating, stromatolites still exist today in places like Shark Bay, Australia. To look at them is to look back through time, seeing structures almost identical to those built when the continents were young. The role of cyanobacteria in this story cannot be overstated. They were among the first organisms to master photosynthesis, the ability to use sunlight to split water and generate energy, releasing oxygen as a byproduct. At first, that oxygen was a poison. The early atmosphere was not designed to handle it, and many microbes perished as levels began to rise. But others adapted, and slowly, the world changed. Oxygen began to accumulate, first in the oceans, reacting with iron to form rust-colored deposits, and later in the air, paving the way for entirely new kinds of life. This event, often called the Great Oxidation, was both catastrophic and creative. Entire ecosystems likely collapsed, yet without it, we would not be here. The very air you are breathing right now exists because tiny microbes billions of years ago learned to turn sunlight into energy. It is a humbling reminder. Our lives are built on the patient labor of organisms so small that you could fit millions of them on the head of a pin. And yet, here's the subtle irony. While oxygen is now essential for complex life, it also brought new dangers. Oxygen is reactive. It damages molecules, causes rust, and even in your body today, it can harm cells if not carefully managed. Early organisms had to invent new ways to protect themselves from oxygen's toxicity, leading to the evolution of antioxidants and specialized enzymes. In other words, the problem and the solution grew together, hand in hand. But let's not rush too quickly. Before photosynthesis reshaped the planet, the earliest cells still had to survive in a world without oxygen. What did that look like in practice? Imagine a shallow pool near a volcanic vent. Warm water, rich in dissolved minerals, constantly stirred by heat. Small molecules drift in, collide, and occasionally form slightly larger structures. Inside a bubble of fatty molecules, a strand of RNA manages to copy itself a few times before breaking apart. Most attempts fail, but sometimes enough copies are made that the bubble divides, carrying some strands with it. It is fragile, precarious, but across millions of years, it becomes slightly less fragile, slightly more efficient. This slow accumulation of advantages, 0.01% at a time, is what carved the path toward life as we know it. It is easy to overlook the sheer patience required. For billions of years, life did not change much. No big leaps, no dramatic transformations. Just endless repetition of microbial lives, reproducing, dying, leaving traces in stone. To us it feels dull, but to nature, it was everything. Stability was the stage on which later complexity could eventually appear, and in that stability, one of the greatest transformations was quietly underway: the collaboration of molecules. No organism is truly self-sufficient. Even the simplest cell relies on networks of reactions, with different molecules supporting one another. Think of it as a village rather than a lone wanderer. RNA working with proteins, membranes providing shelter, minerals offering surfaces. All these pieces together created resilience that no single piece had on its own. Some scientists even argue that the story of life is less about individual molecules and more about systems. Once a network of reactions becomes stable enough to sustain itself, it begins to resemble life, even if no part of it is particularly impressive on its own. It is the combination, the way the parts reinforce each other, that creates something new. And so, we are left with an unresolved picture. We can point to stromatolites, to isotopic fingerprints, to ancient rocks scarred by microbial activity. We can reconstruct how oxygen changed the planet, how cyanobacteria thrived, how the first ecosystems flourished in shallow seas. But we cannot yet say with certainty how the very first cell assembled itself, or why Earth, among all the planets in the solar system, managed to host that delicate balance. As you think about this, perhaps lying back in the quiet of your room, consider how different our world might have been. If cyanobacteria had never mastered photosynthesis, would Earth still be a planet of microbes, its air suffocating to us, its oceans green with bacteria but devoid of fish? Would intelligence ever have emerged, or would the story of life have ended in stillness, too fragile to expand? By the time life had settled into its microbial routines, Earth itself was undergoing constant transformation. Continents were not fixed in place as they appear to us now. They drifted, collided, and split apart, dragging shallow seas and coastlines with them. Volcanoes reshaped the air, meteorites occasionally struck, and yet through it all, life endured. What is perhaps most remarkable is not the fragility of early organisms, but their resilience. Once life had begun, it clung on, adapting to conditions that today would seem utterly hostile. To understand this resilience, you have to look at the way microbes interacted with their environment. Early Earth was filled with energy sources that seem alien to us: sulfur vents, iron deposits, hydrogen gases leaking from rocks. Microbes learned to exploit these, each finding a niche where it could survive. Some stripped electrons from sulfur compounds, others relied on methane. These processes seem technical, even dull, but they mattered enormously. They allowed life to spread into nearly every environment available. Even today, scientists study extremophiles, organisms that live in boiling hot springs, acidic pools, or miles beneath the ice to gain clues about what early life might have been like. For instance, microbes in Yellowstone National Park thrive in water so hot that you can see steam rising from it. Others survive without sunlight, buried deep in rock, feeding only on the faint trickle of chemical energy released as minerals slowly react. These modern survivors are not relics but reminders. The strategies of early life still exist, still working, billions of years later. One of the central questions in understanding life's origin is whether these survival strategies came before or after the ability to harness sunlight. Did photosynthesis evolve late, as a clever adaptation, or was it one of the original tools of life? Evidence suggests that primitive forms of photosynthesis may have appeared very early, long before oxygen filled the air. These early systems did not split water, but used simpler chemicals like hydrogen sulfide. They were inefficient compared to the photosynthesis plants used today, but they may have laid the groundwork for the later revolution that flooded Earth with oxygen. This gradual buildup of complexity is what makes the story of life's origin so different from the stories of mythology. There is no single day when the universe declared, "Now there is life." Instead, there is a series of tiny steps, molecules cooperating, cells experimenting, energy sources shifting that collectively gave rise to a system capable of enduring. It is messy, imprecise, and unsatisfying to those who crave neat beginnings. But it is honest to the evidence. And the evidence is often quiet, a layer of ancient sediment containing unusual chemical ratios, a band of iron-rich rock in Canada or South Africa that speaks of oxygen binding to iron in the oceans, a fossilized stromatolite ridge in Australia. None of these would catch your eye if you stumbled across them on a walk. They are dull stones, weathered lumps, faded streaks of color, but taken together, they are the script of life's first acts, written not in words but in chemistry. Perhaps one of the most fascinating developments in early life was the emergence of cooperation. Modern biology often emphasizes competition, the struggle for survival, the red in tooth and claw picture of evolution. But at the microbial level, cooperation may have been just as important. Cells learned to trade resources, to form biofilms, where one group's waste became another's food. This simple recycling created communities more robust than any lone cell. In a way, it was the first glimpse of what would later become ecosystems. Imagine a shallow lagoon 4 billion years ago. On the surface, bubbles of methane rise from microbial mats. Just below, other microbes strip electrons from sulfur, releasing faint energy. Still deeper, in oxygen-poor mud, still others feed on the leftovers. Each group depends on the others, though none is aware of it. Out of these invisible partnerships came stability. the kind of stability that allowed life to weather the chaos of a young planet. Another turning point came with the gradual increase of oxygen, often referred to as the Great Oxidation. At first, oxygen was scarce, appearing only locally around photosynthetic microbes. But as it accumulated, it began to change everything, altering minerals, reshaping the chemistry of oceans, and forcing life to adapt. For countless organisms, oxygen was toxic, and many went extinct, but others learned to use it, and in doing so, gained access to a much more powerful way of extracting energy from food. Oxygen metabolism was like trading in a bicycle for a rocket. It allowed organisms to produce far more energy, opening the door to greater complexity. Still, this was not a swift revolution. Oxygen levels rose and fell, often for hundreds of millions of years at a time. Scientists believe there were periods when oxygen levels spiked, only to crash again, plunging life back into anoxic conditions. These fluctuations shaped evolution in ways we are still piecing together. It is as if the planet itself was testing life, swinging between extremes, forcing adaptation again and again. And through all this, life's footprint remained microscopic. For more than 2 billion years, the world belonged to single-celled organisms. This can feel disappointing when we imagine the grandeur of dinosaurs or the diversity of the Cambrian explosion. But consider what those billions of years accomplished. Life perfected the basics. It learned to copy itself more reliably. It learned to cooperate. It learned to harness sunlight and oxygen. By the time complexity appeared, the groundwork had been laid so thoroughly that evolution could suddenly accelerate. You and I are the beneficiaries of that patience. Every neuron firing in your brain, every beat of your heart, relies on cellular machinery that was perfected in those early ages. The mitochondria in your cells, which generate energy, descend from ancient bacteria that learned to use oxygen with breathtaking efficiency. The membranes that protect your cells echo the bubbles that first enclosed RNA strands billions of years ago. Even the genetic code itself, the sequence of nucleotides that spells out instructions for life, was largely fixed during that microscopic age. It has remained essentially the same ever since. When you think about this, it reframes our place in the story. Humans are recent, fragile, and temporary, but microbes, those patient builders of stromatolites, those quiet recyclers of elements, are nearly eternal. They shaped the world we inherited, and they remain its foundation. Your own body contains more microbial cells than human ones. The air you breathe, the food you eat, the soil beneath your feet, all depend on microbial processes established billions of years ago. And so, the origin of life is not just a question about the past, it is also about the present. Every moment of your existence is tied to ancient solutions discovered by chemistry long before there were animals or plants. Without those dull but persistent patterns, there would be no forests, no oceans filled with fish, no human societies asking questions about their origins. As you drift deeper into this thought, ask yourself, if life could begin with such simple ingredients, carbon, hydrogen, minerals, and sunlight, does that mean it is likely to appear elsewhere in the cosmos? Or was Earth's balance of conditions so rare, so precise, that it remains a singular miracle? By now, the story of life on early Earth has given us microbes clinging to rocks, bubbles of chemistry, experimenting with survival, and the slow rise of oxygen. But there is another piece of this puzzle that often goes unnoticed, partly because it feels less dramatic than explosions or extinctions. That piece is time immense, unyielding time, the kind of time that grinds mountains down to sand, that lets oceans fill and empty, that allows chemistry to stumble into complexity, not through brilliance, but through repetition. You and I live in a world that measures progress in hours and days. We grow restless if our devices take more than a few seconds to load. Imagine instead a world where progress is measured in millions of years. That was the tempo of early life. Every small advance, a slightly more stable RNA strand, a slightly stronger membrane, a more efficient way to gather energy, took not years, not centuries, but vast spans of geological time. It was like watching grass grow, only stretched to an unimaginable scale. The importance of time becomes clear when we consider one of life's most critical milestones, the invention of the genetic code. Today, every living thing shares the same basic language of life. DNA or RNA made of 4 nucleotides that spell out instructions. What is remarkable is not only that this system exists, but that it is nearly universal. The same code that works in bacteria also works in your cells. Scientists sometimes call this frozen accident, meaning that once the system was established, it became locked in, passed down through every lineage, because any radical change would have destroyed the organisms using it. But how did such a code appear in the first place? That question remains one of the greatest mysteries. Some theories suggest that simpler codes existed first, perhaps with fewer nucleotides, and gradually expanded. Others propose that the code was shaped by chemical affinities between amino acids and nucleotides, meaning the mapping was not random but influenced by natural tendencies. Whatever the case, it is clear that this process took immense amounts of time, with trial and error stretched across countless generations of primitive cells. To appreciate how slow this was, consider that for nearly 2 billion years, life on Earth remained single-celled. Imagine that half of Earth's history spent with nothing more advanced than bacteria. This can feel almost disappointing, as though evolution was dawdling. But during this long span, life was refining its core toolkit: genetic information, energy metabolism, membranes, and the ability to adapt. Once those basics were perfected, complexity could finally unfold. This brings us to another subtle but essential factor: stability. Life needed stretches of relative calm to refine itself. Yes, meteorites struck and volcanoes erupted, but overall, Earth offered long windows of stable conditions where evolution could experiment. Compare this with other planets in our solar system. Mars, for instance, may once have had liquid water and perhaps even the ingredients for life, but its thin atmosphere allowed that water to escape, and its surface grew cold and hostile. Venus, on the other hand, spiraled into a greenhouse effect so intense that its surface now burns hot enough to melt lead. Earth's stability was not inevitable. It was fragile, balanced by a combination of distance from the sun, presence of a protective magnetic field, and cycles that regulated temperature. Without this balance, those billions of years of patient evolution might never have been possible. Even with stability, the environment was not gentle. Ultraviolet radiation from the sun bombarded the surface, lightning storms raged, and the young Earth spun faster on its axis than it does today, making days much shorter. For early microbes, this was not a paradise. It was survival against constant stress. Yet, paradoxically, that stress may have been crucial. Harsh conditions forced life to adapt, to become more resilient, to discover solutions it might not have needed in a calmer world. One of those solutions was diversification. Instead of one pathway for survival, microbes explored many. Some became specialists in extreme heat, others in acidity, others in salt. This branching of strategies gave life resilience. If one environment collapsed, others still supported life. It is the same principle that makes ecosystems robust. Today, diversity spreads risk. This pattern also speaks to something larger. Life is not just chemistry. It is chemistry that learns, not consciously, not with intent, but through variation and persistence. Each failure left behind no trace, but each tiny success, however modest, carried forward, refined by repetition. Over billions of years, these successes stacked up until life reached a tipping point. And here, we touch upon one of the most humbling truths. Complexity did not appear because it was destined to. It appeared because time allowed enough experiments for improbable outcomes to eventually occur. Given billions of years, molecules that rarely form will form. Reactions that rarely sustain themselves will sustain. Systems that rarely persist will persist. Time is the invisible partner in the origin of life, the patient force that turned improbable into inevitable. But was it inevitable? That is the question scientists still debate. If we rewound Earth's history and let it play out again, would life always appear? Would it always settle on DNA, RNA, and proteins? Or was our story the result of chance events, meteorites delivering certain molecules, specific minerals catalyzing key reactions, rare environments providing shelter? If so, perhaps Earth is unique, not because the universe lacks chemistry, but because the dice rolled in a particular way here and nowhere else. There is no definitive answer. What we know is that life, once begun, became incredibly difficult to extinguish. It survived meteorite impacts, ice ages, oxygen poisoning, and global upheavals. That resilience suggests that while the origin may have been delicate, the continuation was robust. Once the pattern of replication and adaptation was established, it proved almost unstoppable. As you think about this, perhaps while your mind drifts in the quiet of night, consider how much of your own body is shaped by time. The hemoglobin that carries oxygen in your blood has its roots in ancient proteins. The mitochondria in your cells are descendants of bacteria that once lived freely. Even the structure of your DNA reflects choices made billions of years ago, locked in by repetition and survival. You are not separate from the deep past, you are a continuation of it. And so the question lingers. Was life inevitable, given enough time and chemistry? Or are we the rarest of accidents, a fragile outcome that might never happen again? If you look at the history of life as a story, there is a moment where the narrative shifts from the simplicity of single actors to the complexity of collaboration. For billions of years, life existed as single cells, bacteria-like organisms that managed everything within their own walls. They could copy themselves, feed, and adapt, but they remained solitary, each cell living and dying on its own. And then, at some point in Earth's deep history, something astonishing happened. Cells began to join forces in a way that would transform the planet forever. This turning point is often described through one of the most profound theories in biology, endosymbiosis. The idea is both simple and radical. Imagine one cell engulfing another, not to eat it, but to keep it as a permanent guest. Instead of digestion, there was cooperation. Over time, the guest cell and the host became inseparable, each dependent on the other. The descendants of that relationship live inside your own body today. They are your mitochondria, the tiny energy factories in your cells. Without them, you would not be alive. The evidence for this is written directly into biology. Mitochondria carry their own DNA, separate from the DNA in your cell's nucleus. That DNA looks more like bacterial DNA than human DNA. Mitochondria even divide in a way similar to bacteria, splitting in two independently of the cell around them. These clues suggest that billions of years ago, a larger ancestral cell swallowed a bacterium capable of using oxygen to produce energy. Instead of destroying it, the cell kept it, turning it into a permanent power source. That partnership was revolutionary. Suddenly, cells had access to vastly more energy than before, and with energy came the possibility of complexity. This was not the only time endosymbiosis occurred. The chloroplasts inside plants, which capture sunlight for photosynthesis, also have their own bacterial ancestry. They too were once independent organisms that took up residence inside a host. When you look at a forest, a field, or even a houseplant, you are seeing the legacy of a microbial merger so successful that it reshaped the planet's atmosphere and created entire ecosystems. But why did this happen only once or twice? If engulfing other cells was such a good strategy, why did it not occur everywhere? The answer is still debated. Some scientists suggest that the odds of such a stable partnership were incredibly small. Most engulfed cells would have been digested as food. Only under rare circumstances would digestion stop and coexistence begin. Others argue that once one lineage discovered this trick, it had such an advantage that it quickly outcompeted everything else. Either way, the result was profound. The emergence of eukaryotic cells, the type of cells that make up all animals, plants, fungi, and eventually humans. Eukaryotic cells were different in more than just energy. They developed internal compartments, separating functions into specialized regions. The nucleus stored DNA. Other organelles managed tasks like protein production or waste processing. This internal complexity was like building a factory with specialized rooms instead of a single, cluttered workshop. The efficiency allowed cells to grow larger, to store more information, and to coordinate more sophisticated processes. It is important to recognize how long this step took. Life appeared on Earth more than 3.5 billion years ago. Eukaryotic cells did not appear until around 2 billion years ago. That means, for over 1 billion years, life remained relatively simple. The leap to complexity was not inevitable. It was rare, hard-won, and dependent on cooperation at the cellular level. When we think about this, it shifts the way we view ourselves. Every breath you take depends on mitochondria, and every leaf you see depends on chloroplasts. Your very existence is not the story of a single lineage, but of ancient mergers, partnerships so intimate that the partners became one. We carry the memory of those mergers in every cell, though most of us never pause to notice it. There is another layer to this story, one that deepens the sense of patience required for life to evolve. Even after eukaryotes emerged, they did not immediately give rise to plants and animals. For hundreds of millions of years, they remained single-celled, floating in oceans, quietly perfecting their machinery. Only later did some begin to cluster together, forming the first multicellular organisms. This too was a kind of cooperation, cells choosing not just to live together but to specialize, some taking on roles of support, others reproduction, others protection. Out of that willingness to divide labor came bodies, tissues, organs. If you think about it, your own body is a continuation of this ancient principle. Your heart cells do not reproduce. Your neurons do not digest food. Your skin cells do not think. Each accepts its role, sacrificing independence for the good of the whole. That trade-off began billions of years ago, when cells first discovered that cooperation could yield stability and power. But before multicellularity took hold, eukaryotes had to endure countless challenges. The Earth itself was not always hospitable. Around 2.5 billion years ago, the planet entered what some scientists call snowball Earth periods, when glaciers may have covered much of the surface. Oceans froze, climates shifted, and life was forced into refuges near hydrothermal vents or beneath layers of ice. These crises could have wiped out fragile new lineages, yet somehow eukaryotes persisted. Perhaps their complexity gave them resilience, or perhaps sheer luck kept them alive in sheltered environments. Whatever the reason, they endured long enough to expand again when conditions eased. And here we see a recurring theme. Life survives by finding balance between chance and necessity. The chemistry of carbon, the possibilities of cooperation, the resilience of simple systems all are necessary. But accidents also shape the path. Rare events like endosymbiosis or climatic shifts that altered the playing field. Without those accidents, the story might have ended differently. For scientists, this raises a profound question. If we search for life beyond Earth, should we expect it to remain simple, like bacteria, for billions of years? Or is complexity eukaryotes, multicellularity, intelligence something that eventually emerges wherever conditions are stable? If endosymbiosis was a rare event, perhaps Earth is unusual. Perhaps elsewhere in the universe, life never moved past the microbial stage. Or perhaps, given enough time, the improbable becomes inevitable. As you lie with this thought, notice how the story of your own body is inseparable from this ancient tale. Every cell in you is a community, a partnership of once separate lives. Every breath you take echoes the moment when a host cell embraced a bacterium instead of digesting it. Every green leaf outside your window carries within it the history of another merger, one that turned sunlight into the energy source for forests and crops. The world you see today is layered with these quiet, improbable collaborations. And so, the origin of life is not just about molecules bumping into each other. It is also about relationships, molecules cooperating, cells merging, lineages intertwining. Without cooperation, Earth might still be a planet of single cells. With it, complexity unfolded, eventually leading to creatures that wonder about their own beginnings. And perhaps that is the question to rest with now. If life depends not just on survival, but on cooperation, what does that say about our own species, built from ancient partnerships, but often forgetting them? When we picture the earliest multicellular organisms, it is tempting to imagine strange creatures, half-formed, drifting in ancient seas. But the beginning of multicellularity was not dramatic. It was subtle, almost invisible. Individual cells simply began to stick together after dividing, instead of separating completely. Over time, this simple clumping evolved into coordinated groups, where cells communicated and divided labor. From those small beginnings came bodies, the first primitive attempts at being something larger than a single cell. The earliest evidence of multicellularity dates back well over 1 billion years, though the fossil record is faint. Some of the oldest known fossils show patterns that resemble mats of cells arranged in sheets. These were not animals or plants as we know them, but experiments in cooperation. Different groups of organisms tried out different strategies. Some formed filaments, others hollow spheres. A few began to show specialized structures, where some cells focused on reproduction, while others handled feeding or structural support. It was the beginning of differentiation, the division of tasks that still defines your own body today. At first glance, it might seem obvious that multicellularity is superior to being a single cell. Bigger is better, after all, because larger organisms can avoid predators, store more energy, and access new ecological niches. But the truth is more complicated. Being multicellular carries costs. Cells in a group must give up some independence. They cannot all reproduce, or chaos would ensue. Some must specialize and trust the whole organism to carry their lineage forward. That means cooperation and sacrifice concepts that nature enforces through evolution. Think of your own body as an example. You contain trillions of cells, but only a small subset are capable of producing the next generation. All the others labor for a purpose they will never experience directly. Skin cells die and slough off. Red blood cells circulate for a few months before being replaced. Neurons live for decades but will never divide. Each plays its role, contributing to the survival of the whole. This system only works because natural selection has favored organisms where cooperation outweighed selfishness. The balance is delicate. If cells go rogue, refusing to cooperate, dividing uncontrollably, you get cancer. A breakdown of the social contract within the body. That fragility reminds us how astonishing it is that multicellularity succeeded at all. The rise of multicellularity reshaped Earth's ecosystems. Suddenly, organisms could grow larger, shaping the environment around them. Sheets of microbial mats gave way to soft-bodied creatures. These were not yet animals in the sense we imagine, but they were more than single cells. They absorbed nutrients in new ways, created habitats for other organisms, and began to alter the chemistry of their surroundings. Fossils from this era, sometimes just faint impressions in ancient rock, show branching patterns, fronds, and quilt-like textures that hint at forms unlike anything alive today. This period in Earth's history is often overshadowed by the later explosion of visible life, but it was here that the foundations were laid. Multicellular organisms experimented with form and function, learning if we can use that word to coordinate growth, communication, and reproduction on a new scale. It was a slow, painstaking process. For hundreds of millions of years, these organisms remained relatively simple, confined to oceans, their bodies fragile and small. But the principle of cooperation had taken root, and evolution had a new canvas on which to paint. One way to think about this is as an increase in information. A single cell contains a genome, but a multicellular organism contains not just genetic information, but organizational information, rules about which genes should be turned on in which cells, when they should divide, and how they should respond to signals from neighbors. This invisible architecture of regulation is what allows a seed to grow into a tree or a fertilized egg to grow into you. The origins of that architecture lie in those early multicellular experiments. Consider how mundane the mechanisms were. Cells used chemical signals, molecules released into their environment to communicate. They built proteins to stick together, forming adhesion molecules like glue. They evolved ways to sense density, recognizing when they were surrounded by neighbors. Each step was small, almost trivial, but together, those steps built the scaffolding for complexity. It was not a leap, but an accumulation of boring, incremental changes, each one significant because of what it enabled next. Multicellularity arose more than once in Earth's history. It appeared independently in animals, plants, fungi, and algae. That suggests that once certain thresholds were crossed, once cells had the tools for adhesion, communication, and gene regulation, the path toward cooperation was tempting enough to be tried again and again. And yet, the specific forms it took varied widely, shaped by environment, by available resources, by sheer contingency. The multicellularity of a mushroom is not the same as that of a jellyfish or a tree. Each is a different solution to the same challenge. How do individual cells surrender just enough autonomy to make the group thrive? As we reflect on this, notice how it mirrors human societies. Groups of people come together, each with their own interests and needs, yet they find ways to cooperate, specialize, and form structures larger than themselves. Sometimes those structures succeed and endure. Other times they collapse under selfishness or imbalance. Nature, it seems, has been wrestling with these themes since the dawn of multicellularity. For scientists studying the origins of life, this stage is especially compelling because it shows how complexity emerges from simplicity without any guiding hand. No one told those cells to stick together. No blueprint directed them. Random mutations and selective pressures shaped them slowly, over vast timescales, until patterns of cooperation emerged. It is a reminder that what feels inevitable in hindsight was anything but certain in the moment. The beauty of this story lies in its patience. Imagine oceans stretching out for millions of years, filled with single-celled organisms experimenting with ways to exist together. Most attempts failed. Most lineages left no trace. But a few succeeded, and their descendants cover the Earth today in every form from towering sequoias to human beings. You are, in a very real sense, the product of a billion-year experiment in cooperation. So as you listen to this, you might ask yourself, if multicellularity arose from the willingness of cells to share, what other possibilities might exist in the universe? Could there be worlds where cooperation took different forms, where organisms built entirely new ways of living together? Or is the story of Earth a slow march from solitude to community, something universal? When we think about the story of life, one of the most dramatic turning points came with the appearance of photosynthesis. Before it, Earth's biosphere was limited to chemical pathways that were clever but narrow, feeding on molecules that happened to be available. After it, the planet was transformed. With photosynthesis, organisms could harvest energy directly from sunlight, the most abundant resource of all. That shift did not just fuel new kinds of life, it changed the entire atmosphere, reshaping the environment in ways that would ripple forward through billions of years, eventually setting the stage for creatures like you. The earliest photosynthesizers were not plants. Plants would not appear until much later. Instead, they were microbes, tiny cells floating in the ocean, some of them belonging to groups we now call cyanobacteria. They discovered a way to use pigments to capture light energy and funnel it into chemical reactions that powered their survival. At first, these systems were clumsy. The earliest forms of photosynthesis did not release oxygen. They relied on other molecules like hydrogen sulfide, abundant in ancient oceans, to donate electrons and make the reactions work. But over time, one lineage of cyanobacteria evolved a method of using water as the electron donor. That small innovation had enormous consequences, because when you split water, you release oxygen. At the time, Oxygen was not the life-giving gas we take for granted. It was a poison to most of the organisms alive then. They had evolved in a world where oxygen levels were almost non-existent, relying on chemical reactions that worked only in anoxic conditions. For them, the spread of oxygen was catastrophic. What you know as the Great Oxidation Event was, for countless ancient microbes, an extinction. The oceans rusted as iron reacted with oxygen and precipitated out as vast deposits of banded iron formations, some of which you can still see preserved in ancient rocks today. The sky itself changed, with new atmospheric chemistry altering the color of sunsets and the behavior of storms. It is easy to overlook just how slow this process was. To you, it may sound like a sudden revolution, but it unfolded over hundreds of millions of years. Oxygen built up first in local patches, then in the shallow oceans, and only eventually in the atmosphere. The tipping point was not a single day or year, but a gradual accumulation, like filling an immense vessel drop by drop. Yet the consequences were permanent. Once oxygen was part of the atmosphere, it created both dangers and opportunities. The dangers were clear. Oxygen is reactive. It breaks down molecules that cannot withstand it, damaging proteins and DNA. Organisms that had thrived in oxygen-free environments suddenly found themselves under stress. Many vanished, driven into hidden niches where oxygen could not reach, like deep sediments or hydrothermal vents. But for others, oxygen became a new tool. With it, cells could perform a kind of respiration far more efficient than anything possible before. Instead of eking out energy in small amounts, they could unleash powerful chains of reactions that produced many times more energy per molecule of food. That surplus of energy became the fuel for complexity. Think of it this way. If life were running on candles before, oxygen allowed it to switch to electric lights. With more power came the possibility of building larger, more complex organisms. Energy that once was scarce could now be spent on movement, on building structures, on developing internal systems. The path to animals, plants, and fungi was opened not by some sudden act of invention, but by this chemical shift in the very air and water of the planet. Cyanobacteria were the unlikely authors of this transformation. Small, simple cells, still abundant today in ponds, oceans, and even on rocks, quietly rewrote the script of life. When you see a greenish film on the surface of stagnant water, you are looking at the descendants of the organisms that made the modern Earth possible. Their work did not end billions of years ago. They continue to produce a significant portion of the oxygen you breathe, working tirelessly, unnoticed, across the globe. From a human perspective, this raises a fascinating point. Life is not just shaped by the environment, it reshapes it. Before photosynthesis, Earth was a planet of chemistry, its atmosphere a reflection of geology and volcanism. After photosynthesis, biology began to dominate. Living things started to regulate the balance of gases, influencing temperature, weather, and even the distribution of minerals. The Earth we know, the one with blue skies, breathable air, and stable climates, exists because life itself made it that way. And yet, for all the grandeur of this transformation, the details are surprisingly mundane. The pigments that made photosynthesis possible, like chlorophyll, are simple molecules compared to the complexity of life itself. The metabolic pathways that process light energy are just chains of chemical steps, each one unremarkable on its own. But string them together, and you get the power to sustain civilizations of microbes, to flood the atmosphere with oxygen, to sculpt the destiny of the biosphere. It is a reminder that history is often driven not by grand gestures, but by the accumulation of small, repetitive actions. In studying this event, scientists face challenges. Rocks from that era are scarce and altered by time, so evidence is fragmentary. Geochemists piece together clues from isotopes, from mineral deposits, from faint chemical signatures locked in stone. Each discovery adds a sliver to the puzzle, but much remains uncertain. Did oxygen build up in fits and starts, rising and falling before stabilizing? Were there multiple waves of oxidation, separated by long intervals of stagnation? The answers are still debated. showing that even for an event so fundamental, our understanding remains incomplete. This incompleteness is not a flaw but part of the beauty of the story. The origin of photosynthesis and the Great Oxidation Event are like distant echoes. We can hear them faintly but cannot see the source directly. Our reconstructions are careful, tentative, filled with room for revision. And yet, the overall message is clear. Life has always been both fragile and powerful, able to alter the planet on timescales far beyond our imagination. As you listen, perhaps you might reflect on the irony. The oxygen you are breathing right now, flowing silently into your lungs, was once a deadly pollutant. It killed untold numbers of organisms long before humans existed. What gives you life today once caused mass extinctions. This dual nature oxygen, as both poison and necessity, captures something essential about the origin of life. Progress is rarely clean. Every step forward is shadowed by loss. So we might ask, if life on Earth reshaped the planet in this way, what might happen on other worlds? Could alien biospheres have their own versions of oxygen events, where chemistry and biology clash and then reconcile? Or is this story unique to our small blue planet, an accident of evolution that cannot be repeated? One of the most mysterious transitions in the history of life was the origin of complex cells. Up until this point in the story, all organisms were simple. They were what we call prokaryote cells without a nucleus, without the intricate compartments you might remember from high school biology diagrams. They were efficient, resourceful, and incredibly adaptable, but they were also limited. Complexity requires space, organization, and new ways of managing energy. For billions of years, life managed without those things. Then, slowly, something extraordinary happened. The first truly complex cells, what we call eukaryotes, appeared. Unlike their prokaryotic ancestors, they had internal membranes that folded into specialized compartments. They had a nucleus to house their DNA, separating genetic information from the rest of the cell's activity. They had elaborate structures for processing energy and coordinating tasks. In essence, they became tiny cities, with roads, factories, and power plants working together in astonishing harmony. The question is, how did this leap occur? For decades, scientists puzzled over it. The eukaryotic cell seemed too complicated to have appeared step by step. Its parts depend on each other so tightly that imagining an intermediate form feels difficult. Yet evidence, gathered slowly through both fossil records and molecular biology, suggests an elegant solution: cooperation. The story is often told through the theory of endosymbiosis. Imagine, if you will, a large ancient cell engulfing a smaller one. Normally, this would be a meal the smaller cell digested and broken down for nutrients. But in some cases, instead of being destroyed, the smaller cell survived inside its host. It kept its own machinery, its ability to generate energy. Over time, the relationship shifted. The engulfed cell provided energy, while the host offered protection and stability. They stopped being predator and prey and became partners. This partnership left traces that are still visible today. The mitochondria inside your cells, those tiny energy-producing organelles, were once free-living bacteria. They still carry their own DNA, distinct from the DNA in your nucleus. They divide on their own, almost like miniature organisms within you. If you look closely, you can see their bacterial ancestry in their genes, in their membranes, even in the way they reproduce. Chloroplasts in plants tell a similar story. They too were once free-living cyanobacteria, captured and domesticated by an ancient eukaryotic ancestor. It is difficult to overstate how radical this was. Endosymbiosis was not just a quirky accident, it was a revolution. By gaining mitochondria, cells suddenly had access to vastly more energy. Each mitochondrion works like a tiny powerhouse, pumping out ATP, the molecule that fuels life's activities. With that extra energy, cells could afford to build larger genomes, more complex structures, and new systems of communication. Energy that had once been scarce now flowed more freely, supporting experiments in complexity that simple cells could not manage. The nucleus, too, was transformative. By separating DNA from the bustling activity of the cytoplasm, cells could regulate gene expression in more sophisticated ways. They could fine-tune when and where proteins were made, coordinating responses to the environment with new precision. Internal membranes folded into endoplasmic reticula and Golgi apparatus, processing proteins and lipids, shipping them to the right places. The cell became not just a bag of molecules, but a coordinated factory, capable of producing and organizing at scales never seen before. The fossil record offers faint glimpses of this transition. Microfossils, preserved in ancient rocks, show larger cells with internal complexity, emerging more than 1.5 billion years ago. But fossils are rare and ambiguous. Much of what we know comes instead from comparing DNA sequences, tracing the evolutionary history of genes. These studies show that eukaryotes are chimeras, their genomes a mixture of contributions from archaea-like ancestors and bacteria-like partners. Life's family tree is not a neat branching diagram but a web, with threads of symbiosis woven into its structure. And here, again, the pace was slow. Eukaryotes did not appear overnight and immediately take over the planet. For hundreds of millions of years, they remained relatively rare, coexisting with prokaryotes without overshadowing them. It took time for the advantages of complexity to accumulate, for ecosystems to adjust, for innovation to spread. In a sense, life tested the waters cautiously before embracing this new architecture fully. You might find it humbling to consider that every cell in your body carries this history. Your mitochondria, still bearing echoes of their bacterial past, churn out energy every second of your life. Without them, you could not think, move, or even exist. Each breath you take feeds oxygen to them, and they, in turn, feed energy to you. You are, at the most literal level, a living collaboration between ancient organisms that chose not to kill each other but to live together. That cooperation was not inevitable. Countless engulfed cells must have perished in failed attempts before one partnership worked, but once it did, it opened doors that would never close again. It shows that evolution is not just about competition. Sometimes, survival comes from alliance, from sharing rather than devouring. That principle, first written in the language of cells, echoes through the rest of life's history, in symbioses between fungi and plants, corals and algae, even humans, and the microbes of our own gut. This raises a philosophical question. What does it mean to be an individual if the very foundation of your being is a merger? If your cells are built from parts that once belonged to entirely separate creatures, is individuality an illusion? Or is it, instead, a reminder that life is defined not by rigid boundaries but by networks of cooperation? Science may never reconstruct every detail of how the first eukaryotic cells formed. The evidence is too scattered, too ancient, but the broad strokes are clear enough. A rare and improbable event reshaped the possibilities of life. Without it, Earth might still be a world of simple microbes, never progressing to animals or plants. The leap to complexity was not guaranteed. It was a fragile, uncertain step, one that might not happen on other worlds. Which leaves us with a haunting question. If we find life elsewhere in the universe, will it be stuck in simplicity, or will it have found its own path to complexity? Once eukaryotic cells had established themselves, the stage was set for the next great transformation: multicellularity. The journey from single-celled eukaryotes to complex multicellular organisms was neither sudden nor straightforward. It was a gradual, painstaking process, one that required innovation, patience, and above all, cooperation. To understand this step, it is helpful to imagine the oceans of that era, vast, shallow seas filled with microscopic eukaryotic cells, each navigating survival in a world still dominated by simpler prokaryotes. Early multicellular life likely began as colony cells that remained attached after division. At first, these colonies were simple, a handful of cells forming clusters with no specialization. Each cell carried out the same functions it had as a single entity, absorbing nutrients, reproducing, and responding to environmental cues. Yet over time, Subtle advantages emerged when cells began to differentiate. Some specialized in capturing food, others in reproduction, and some in structural support. That division of labor was the key to scaling up, to building more complex forms capable of surviving and thriving in diverse environments. The first multicellular organisms were tiny and fragile. Fossil evidence, though rare, shows impressions of filamentous forms and sheets of cells clinging together, stretching across shallow waters. These organisms were not predators or sophisticated grazers. They were more like living mats, absorbing energy and nutrients directly from the environment. Yet even in their simplicity, they represented a radical innovation, cooperation at a scale never seen before. A single cell, acting alone, has limits, but many cells working together can overcome environmental challenges, survive fluctuating conditions, and exploit new niches. Multicellularity required more than just physical adhesion. Cells had to communicate, coordinate growth, and regulate gene expression across the group. Chemical signaling pathways evolved to transmit information from one cell to another, ensuring that tasks were divided efficiently. Imagine tiny chemical messages traveling through a colony, instructing some cells to divide while others halt, some to absorb nutrients while others produce protective structures. These chemical conversations were the first whispers of what would become tissues and organs in later organisms. One of the first major experiments in multicellularity occurred in simple algae and protofungi. In these early communities, cooperation was fragile. Cheating cells that refused to cooperate could undermine the colony. Evolution solved this problem in several ways. Some colonies evolved mechanisms to police cheating, eliminating non-cooperative cells. Others restricted reproduction to specific cells, ensuring that the cooperative framework remained intact. These strategies, refined over millions of years, laid the groundwork for the sophisticated social contracts that exist in all multicellular life today. It is remarkable to consider that the very principles shaping your own body, the division of labor among cells, the specialization of tissues, the coordination of complex functions, were first tested in these tiny, humble colonies. Your heart does not pump, your neurons do not process food, your skin does not reproduce. Each part of your body relies on the others, a direct legacy of these early experiments in cooperation. Life, it seems, has always been a story of interdependence. Multicellularity opened doors for innovation. With more energy and organizational capacity, organisms could grow larger, explore new environments, and develop structures to exploit sunlight, nutrients, and shelter more efficiently. It also set the stage for predator-prey dynamics, an evolutionary driver that would intensify selection pressures and accelerate complexity. Life was no longer only about surviving chemical gradients or absorbing nutrients. It began to interact, compete, and influence its surroundings on a larger scale. The fossil record provides glimpses of these changes, though evidence is sparse. Impressions of simple multicellular organisms date back over 1 billion years, but more recognizable forms appear later, during periods when oxygen levels had stabilized enough to support larger, energy-demanding bodies. By this time, colonies had begun experimenting with shapes, layering cells into sheets, spheres, and filaments. These structures were not just functional, they were adaptive, shaped by selective pressures in their environments. Some forms enhanced nutrient absorption, others provided protection, and some optimized reproduction. Evolution was quietly sculpting the first truly complex organisms. This era also underscores the importance of environmental context. Stable, oxygenated waters provided the energy needed to support multicellularity. Chemical nutrients, sunlight, and shallow coastal environments created opportunities for growth and experimentation. Fluctuations in these conditions acted as selective filters, shaping which colonies survived and which faded. Multicellularity did not spread uniformly across the planet. It emerged where conditions allowed, gradually radiating into diverse forms. From a broader perspective, multicellularity illustrates a recurring theme in the history of life: incremental change, driven by cooperation. None of these early colonies were immediately spectacular. They were unassuming, fragile, and often overlooked by the fossil record. Yet their significance cannot be overstated. Each small innovation, adhesion, signaling, differentiation, was a stepping stone toward the astonishing diversity of life that would follow, from corals and sponges to forests, insects, and eventually humans. In contemplating this history, one realizes that the roots of complexity are not in sudden leaps, but in subtle, patient processes. Evolution does not rush. It experiments endlessly, discards failures, and amplifies small advantages over unimaginable spans of time. The first multicellular colonies were humble experiments, and yet they set the stage for ecosystems, bodies, and entire biospheres built on cooperation. And so, as you consider your own existence, remember that the patterns of life inside your body, your specialized cells, your organs, your tissues, are direct descendants of these ancient experiments. Multicellularity was not just a step toward animals or plants. It was a fundamental reimagining of what life could be: organized, cooperative, and capable of sustaining complexity far beyond the limits of a single cell. One lingering question remains. If multicellularity required so many rare steps, adhesion, communication, specialization, how common might it be elsewhere in the universe? Could alien life follow a similar trajectory? Or are we witnessing a rare alignment of chemistry, energy, and time? Perhaps other worlds have colonies of simple cells experimenting silently in their oceans, waiting for the rare moment when cooperation tips the scales toward complexity. As multicellular life slowly took hold, the next great challenge for organisms was not merely survival, but thriving in increasingly complex environments. Life was no longer just about holding on to chemical energy or reproducing in simple ways. It had to navigate competition, predation, and environmental fluctuations. These pressures would drive one of the most remarkable innovations in Earth's history: the development of body plans, the architecture of life that would eventually allow organisms to move, sense, and interact with their surroundings in entirely new ways. Early multicellular organisms were limited. Many were flat, soft, and simple, relying on diffusion to move nutrients from the outside to the interior. This method worked only at small scales. As organisms grew larger, diffusion alone was insufficient. Cells deep within a body required specialized structures to receive nutrients and expel waste. This limitation created the first real evolutionary pressures to organize internally, to create channels, cavities, and structures that could distribute resources efficiently. It was a subtle but profound challenge: how to build something larger without losing the benefits of being small and simple. One solution came in the form of symmetry. Organisms began to organize their bodies around axes, bilateral symmetry, radial symmetry, and variations in between. Bilateral symmetry, with a clear front and back, left and right, allowed for directional movement, a key advantage for seeking food, avoiding predators, and exploring the environment. Radial symmetry, often seen in modern-day jellyfish and sea anemones, facilitated interaction with the world from all sides, useful for stationary or slow-moving organisms. These early experiments in body organization set the stage for the incredible diversity of forms that would later populate oceans, forests, and eventually land. Alongside symmetry came segmentation, another quiet but powerful innovation. Dividing a body into repeated units allowed organisms to specialize parts for different tasks. One segment could manage movement, another digestion, and another reproduction. Segmentation was not immediately sophisticated, but it provided a flexible framework that evolution could later refine. Worms, arthropods, and even vertebrates would exploit this principle, building increasingly complex and capable bodies from the same fundamental design. The fossil record of this time is patchy but revealing. Soft-bodied organisms left faint impressions, sometimes just sheets of cells preserved in ancient sediment. These fossils hinted experimentation: elongated forms, frond-like structures, simple appendages. Nothing dramatic yet, but enough to suggest that evolution was testing multiple approaches simultaneously. Some lineages would succeed, others would vanish. The pattern is clear. Life advances through trial and error, with countless failures hidden beneath the surface of history. Another subtle driver of change was interaction with the environment. Organisms that could manipulate their surroundings, even slightly, gained advantages. For example, growing in a slightly elevated structure allowed access to light or oxygen, or facilitated filter feeding from water currents. Small movements, changes in form, and adjustments in growth all contributed to survival. These incremental modifications, though invisible over short timescales, accumulate over millions of years into entirely new capabilities. Energy efficiency remained central. Oxygen metabolism, perfected in eukaryotes, now supported these increasingly complex bodies. Cells could power specialized structures like cilia, flagella, and contractile elements to generate movement. They could develop simple sensory structures to detect light, chemical gradients, or vibrations. These early senses were crude, yet they represented the foundation of perception, of interacting with the world in a purposeful way. Without the metabolic energy provided by oxygen, these innovations would have been impossible. At the same time, reproduction became more sophisticated. Simple division was no longer enough for larger organisms. New methods arose: specialized reproductive cells, cycles that coordinated with environmental cues, and mechanisms to protect offspring. These strategies ensured that life could persist across generations, even as bodies became more complex and environments more challenging. Reproduction itself became a site of experimentation, with evolution testing different ways to balance investment in growth, survival, and offspring. Throughout this period, life's complexity was still microscopic by modern standards. Creatures were small, soft, and mostly aquatic. Yet the principles established during this era, symmetry, segmentation, specialized cells, sensory capability, and coordinated reproduction, would underpin all later animal life. From the smallest worm to the largest mammal, these strategies remain encoded in the genetic architecture of modern organisms. Every heartbeat, every step, every sensory perception is built on foundations laid hundreds of millions of years ago. It is easy to overlook the quiet patience of this process. Evolution does not rush. It experiments slowly, discarding failures and preserving successes. Millions of years pass between each meaningful innovation. Yet within these stretches of time, life explores possibilities that human imagination could scarcely conceive. Simple cells adhering together became colonies. Colonies became differentiated tissues. Tissues became body plans, and body plans eventually gave rise to movement, perception, and complex interaction with the environment. It is a story not of sudden invention, but of patient accumulation. The environment itself remained an active participant. Oxygen levels fluctuated, nutrients shifted, and geological events reshaped coastlines and oceans. These external pressures acted as filters, testing which forms could survive and which would vanish. Organisms were not merely passive recipients of conditions. They were active experimenters, adapting in response to challenges. Those that succeeded laid down the principles that would govern later life: cooperation, specialization, energy efficiency, and adaptability. Even today, we can see echoes of this era in living organisms. Flatworms, jellyfish, and simple algae preserve body plans and strategies that date back over 500 million years. They remind us that complexity is not inevitable, but that certain solutions, once discovered, are resilient. Evolution may be messy, but it is also conservative. When something works, it tends to persist, spreading through lineages and shaping the course of life for eons. And so, as you reflect on your own body, your nervous system, your organs, your capacity for movement and perception, you are witnessing the legacy of this quiet experimentation. Every organ system, every coordinated function, is built on the foundations established by organisms long vanished, whose struggles and innovations paved the way for life as we know it. Perhaps the most intriguing question remains: if these principles of symmetry, segmentation, and specialization emerged here on Earth, could they appear elsewhere? Are body plans a universal consequence of multicellularity, or did chance and Earth-specific conditions guide these innovations? The answers remain uncertain, leaving us to wonder about the possibilities of life across the cosmos. As multicellular life experimented with form and function, another quiet revolution was taking place beneath the ocean's surface: the development of predation. For hundreds of millions of years, life had been relatively simple, with organisms absorbing nutrients passively or filtering them from the surrounding water. But once some creatures developed structures capable of actively capturing, consuming, or processing other organisms, the evolutionary landscape changed dramatically. Predation introduced an entirely new set of pressures, reshaping ecosystems and accelerating the pace of adaptation. Predation may seem like a straightforward concept, but in the early oceans, it was radical. For billions of years, life had been relatively benign. Microbes grazed on surfaces, simple multicellular forms drifted passively, and competition existed mostly for space or chemical resources. The emergence of active hunters, organisms capable of moving purposefully toward prey, introduced risk in an unprecedented way. Suddenly, survival depended not only on resisting chemical or environmental stress, but on avoiding being eaten. Evolution had entered a new phase, one defined by interaction, strategy, and arms races. This development triggered what some scientists call an evolutionary escalation. Prey species began evolving defenses, thicker coverings, spiny projections, burrowing behaviors, or faster movement. Predators, in turn, refined their attack strategies, improving sensory capabilities, locomotion, and hunting efficiency. Even subtle changes, a slightly stiffer body, a small appendage for grasping, or a primitive eye spot, could confer a significant advantage. These tiny modifications, over vast timescales, led to a rapid expansion of forms, sizes, and behaviors that we recognize as the foundations of complex animal life. The fossil record from this period, known as the Ediacaran and early Cambrian, is fascinating. Faint impressions in sedimentary rocks reveal strange, often enigmatic creatures. Some appear as simple discs or fronds anchored to the seafloor. Others have segmented bodies, ridges, or tubular structures, hints of defensive or predatory adaptations. The evidence suggests a world teeming with experimentation. Predators exploring movement and capture, prey developing protective structures, and ecosystems growing increasingly dynamic. These were early experiments in survival strategies that would define life for hundreds of millions of years. One striking innovation during this period was the development of sensory structures. To be an effective predator or evade one, organisms needed to perceive their environment. Simple light-sensitive patches evolved into eyes capable of detecting motion or shapes. Mechanoreceptors, able to sense vibration or pressure, allowed organisms to respond quickly to danger or opportunity. Even the most primitive senses represented leaps in information processing, enabling creatures to act rather than react passively to their surroundings. These sensory innovations, while rudimentary, set the stage for the incredible diversity of perception found in later animals, from insects navigating complex visual landscapes to mammals interpreting sound and touch. Alongside sensory evolution came improvements in locomotion. Simple cilia or flagella allowed movement on a small scale, but larger organisms required more coordinated systems. Muscles and contractile fibers developed, allowing flexible, purposeful motion. These advancements were not uniform. Different lineages experimented independently, producing a variety of movement strategies. Undulating bodies, appendages for crawling, or structures for suspension in water. The result was a period of rapid experimentation, where natural selection tested combinations of form, function, and behavior on a planetary scale. Predation also influenced reproduction. Organisms facing high risk of being eaten often favored strategies that increased offspring survival. Some produced large numbers of small, rapidly dispersing gametes. Others developed protective coverings or burrowing behaviors to shield embryos. This period demonstrates how interconnected life's challenges are. A change in one aspect of ecology, predation ripples through physiology, behavior, and reproduction, shaping evolution in unexpected ways. The metabolic demands of predation also reinforce the advantages of eukaryotic energy efficiency. Chasing, grasping, and processing prey required substantial energy, made possible by mitochondria. Here, the ancient symbiotic partnership that created complex cells revealed its significance. Only organisms with sufficient internal energy could sustain these new behaviors, allowing predation to become an effective evolutionary strategy rather than an occasional experiment. It is fascinating to reflect on how such a violent, stressful innovation, hunting, and being hunted, produced a surge in complexity. Predation introduced competition on a new scale, forcing organisms to innovate rapidly. Evolution was no longer only a matter of surviving chemistry or passive growth. It now involved strategy, adaptation, and continuous feedback between species. This dynamic reshaped entire ecosystems, turning the oceans into arenas of interaction and experimentation. Even today, the echoes of these early predatory pressures are visible. Many defensive strategies in modern animals, shells, spines, speed, camouflage, trace back to the same evolutionary arms race that began hundreds of millions of years ago. The first predators were not apex hunters like lions or sharks. They were tiny, simple organisms whose innovations created ripple effects still shaping life. Every predator-prey relationship you observe today is part of a story that began long before vertebrates, long before plants colonized land, long before humans existed. This era also offers a humbling reminder of the contingency of evolution. Predation could have failed as an innovation. Early predators might have been inefficient, their prey too resilient, or environmental conditions unfavorable. But once it succeeded, it accelerated evolution on a scale unimaginable in the preceding billions of years. Complexity, diversity, and specialization all intensified, transforming life from passive collections of cells into active, interactive communities. And yet, as dramatic as predation was, it was still built on patience, iteration, and cooperation at lower levels. Cells still adhered, still communicated, still divided labor within multicellular bodies. These ancient partnerships created the platform for the next steps. Without them, predation as a strategy might never have been possible. Cooperation at the cellular level enabled conflict and competition at the organismal level, a fascinating paradox that echoes through life's history. As you reflect on this, consider the broader implications. Predation demonstrates that evolution is not a gentle process of gradual improvement. It is a complex interplay of cooperation and competition, innovation and risk, success and failure. Tiny, seemingly trivial innovations, a light-sensitive patch, a flexible appendage, a sticky cell adhesion molecule can set the stage for dramatic transformations over millions of years. The ocean of early life was not calm. It was a laboratory, testing every possible combination of form, function, and behavior. So the question lingers: if predation was such a powerful driver of complexity on Earth, what might life look like elsewhere? Could alien worlds experience similar arms races, or might other evolutionary pathways dominate, producing complexity without hunting and evasion? We cannot know, but the story of early Earth reminds us that life's most profound innovations often emerge from pressure, stress, and the need to survive in the presence of competitors. By the time life approached the late Precambrian and early Cambrian periods, the stage was set for what scientists call the Cambrian explosion, a breathtaking, rapid diversification of complex organisms. This was not a sudden event in the sense of hours or years, but in geological terms, over a few tens of millions of years, the world transformed. Soft-bodied, simple multicellular organisms had begun experimenting with forms, movement, and interactions. Now, With oxygen levels stabilized and predation driving evolutionary innovation, life expanded into shapes and strategies that had never existed before. During this period, almost every fundamental body plan recognizable today began to emerge. Worm-like forms developed segmented bodies. Appendages appeared for grasping and swimming, and sensory organs became more sophisticated. Eyes capable of detecting shapes and light gave predators an edge, while prey evolved speed, camouflage, and protective armor. It was a period of rapid experimentation, as though evolution had suddenly pressed a fast-forward button on complexity. The oceans teemed with activity soft-bodied creatures, armored arthropods, primitive mollusks, all exploring the possibilities of size, structure, and behavior. The fossil record, though incomplete, preserves remarkable evidence. Impressions of trilobites, sponges, and early mollusks hint at a world increasingly intricate and competitive. Many lineages would fail, leaving no trace, while a fortunate few established lineages that would persist and diversify over hundreds of millions of years. These survivors laid the groundwork for the ecosystems we know today, layered, interconnected, and filled with specialization. What began as humble colonies of cells had now given rise to ecosystems rich with predator-prey dynamics, niche specialization, and ecological networks. Energy management remained central to this story. Larger, more active organisms required efficient metabolism to sustain movement, sensory processing, and reproduction. The ancient endosymbiotic partnerships that had created mitochondria now proved indispensable. Without sufficient cellular power, these innovations in size, mobility, and complexity would have been impossible. It is a reminder that the smallest, quietest events, like a bacterium entering a host cell, can ripple forward, enabling transformations on a planetary scale. The Cambrian explosion also demonstrates the importance of environmental context. Shallow seas, nutrient-rich waters, and stable conditions created opportunities for expansion. Geographic features like continental shelves provided space for diversification, while fluctuations in climate and sea levels created selective pressures that accelerated adaptation. Life, in turn, reshaped its environment. Burrowing organisms aerated sediments, predators altered population structures, and photosynthesizing organisms continued to regulate oxygen and carbon dioxide levels. The planet became increasingly co-created by its living inhabitants. At this point, it is helpful to pause and consider the scale of innovation. Body plans that remain recognizable today, the arthropods, with their jointed appendages, the chordates with their primitive nerve cords, had their origins in this window of experimentation. Yet the creatures themselves were strange, unfamiliar, and often short-lived. Evolution was testing many possibilities, discarding failures, and preserving those with adaptive advantages. The process was slow and meticulous, but its effects would resonate for billions of years. Meanwhile, reproduction and development became more sophisticated. Simple cellular division was no longer adequate for large, complex bodies. Specialized reproductive cells, embryonic development, and life cycles tailored to environmental conditions became common strategies. Organisms experimented with eggs, larvae, and metamorphosis. These strategies not only ensured survival but also promoted diversification, as different species adapted to different ecological niches. The stage was set for the vast complexity of ecosystems, with interactions between species shaping evolutionary trajectories in ways that continue today. The rise of predation, body plans, and reproductive innovation created a feedback loop. Predators pressured prey to innovate, prey innovations pressured predators, and environmental fluctuations favored those that could adapt most efficiently. This dynamic interplay accelerated evolution in a way that simple, static environments could not. Life had entered a phase of rapid expansion, diversification, and experimentation, producing the intricate ecological webs that define our planet. Human beings, though far removed in time, are products of this deep history. Every organ system, sensory capability, and behavioral trait we possess is built on the foundations laid during these ancient periods. The capacity to move purposefully, perceive the world, process information, and reproduce in complex ways all trace back to innovations in cellular cooperation, energy management, multicellularity, and early sensory development. Every heartbeat, every neural signal, every glance is a continuation of evolutionary experiments that began hundreds of millions of years before humans existed. Yet even in this success, the story is incomplete. Evolution is ongoing, and the processes that shape the Cambrian continue to operate in subtle ways. Microbial life still dominates biomass. Cooperative partnerships between species continue to evolve, and ecological interactions maintain pressure on all life forms. The lessons of the past remind us that complexity is fragile, contingent, and the product of countless small events, accidents, and innovations. One striking implication of this history is the interplay between chance and necessity. Life on Earth could have remained simple for billions of years. Many experiments failed, many organisms vanished without leaving descendants. But certain rare events like endosymbiosis, oxygenation of the atmosphere, and the development of multicellular cooperation created opportunities that propelled complexity forward. The result is the biosphere we see today, layered, interconnected, and filled with organisms capable of astonishing adaptation. The open-ended question that remains is compelling. If life elsewhere follows a similar trajectory, will it necessarily experience periods of rapid diversification, like the Cambrian explosion, or is this phenomenon unique to Earth's particular conditions? Could other planets host life that remains simple for eons, or might they also produce bursts of innovation under the right combination of energy, cooperation, and environmental stability? These are questions we cannot yet answer, but contemplating them emphasizes both the rarity and the universality of life's experiments. And so, as you reflect on the grand arc of life from single molecules to cooperating cells, to multicellular colonies, to predators, and diverse ecosystems, you are contemplating a story that spans billions of years, a story built on tiny patient steps, on quiet chemical reactions, on improbable events that multiplied over unimaginable time. Every feature of modern life, from the energy in your cells to the ecosystems outside your window, is a continuation of this narrative. In the end, the origin and evolution of life teaches us a profound lesson. Complexity emerges from cooperation, adaptation, and the relentless trial and error of natural processes. The world we inhabit today, with its forests, oceans, animals, and humans, is the product of patient experimentation, a series of interconnected events stretching back to the earliest cells. And perhaps the greatest wonder is this: the same principles that guided the first tiny colonies of cells still guide life now, continuing to shape evolution in ways we can barely imagine.